Ganguli. I'm in the Department of Applied Physics at Stanford, and I'm going to talk about mechanistic models of cognition from perception to navigation to semantic development. So I'd like to preface by saying, of course, we're in a golden age of neuroscience at the moment. We have access to large-scale recordings and stimulation data. Advances in machine learning and computation allow us to generate highly accurate veridical models of brain function, at least in terms of predicting the activity of individual neurons. But this raises a profound question about the very nature of explanation in neuroscience. Are we simply replacing things that we would like to understand but don't with something else that we don't understand, i.e. replacing brains with complex veridical models? So statistical and computational neuroscience has a major understanding problem, and the theme of this talk is to develop and apply techniques in explainable artificial intelligence to understand and extract testable hypotheses about accurate but complex neural models. Explainable AI is a set of techniques or a set of goals that aim to try to trace the causal flow of information through circuits and understand how that information flows to mediate function. Okay. So, the outline of the talk, I'll give you 4 very high-level kind of take-home messages of 4 vignettes that we've been working on in my lab. The first at the top is a state-of-the-art model of the retinal response to natural movies. And then we aim to understand how it reproduces 20 years of experiments in artificial stimuli. The second is to derive from first principles the properties of the 4 most dominant retinal ganglion cell types from the structure of natural movies, in the primate. Okay. The third is to try to explain a functional origin for grid cells. And we're going to derive a theory for the obligatory emergence of grid cells from biological plausible neural circuits that path integrate. And the fourth, we'll go even higher level to psychology. We're going to derive a simple theory of how neural circuits can recapitulate diverse psychology experiments, development of semantic categories in infants. Okay, so let's get started. So the work on network models of the retina was done by a very talented group of grad students and postdocs. There's a sequence of papers here which will contain all of the details. So here's a schematic of the retina. It's already a sort of a one hidden layer neural network. Light comes in on the photoreceptors that generates electrical signals that propagate through a layer of bipolar cells laterally connected by horizontal and amacrine cells. And that generates activity in retinal ganglion cells, and we have 1 million optic nerve fibers that go to our brain that tell us everything we know about the visual world. A common model of the retina is a very simple linear nonlinear model consisting of a spatiotemporal linear filter of light patterns propagating through a rectifying nonlinearity. This is a very good model of the retinal response to low-resolution white noise, but it can't accurately get the retinal response to natural movies, the very stimuli that sculpted the evolution of the retina. And of course, it doesn't reflect the underlying complexity of the retina at all. So we decided to try to fit a deep neural network model of the retina that mimics the structure of the retina. So it takes as input stimuli, generates retinal ganglion cell outputs, and we try to adjust the synaptic weights of this neural network so it mimics the retinal response of salamander retinal ganglion cells while they're exposed to natural movies. This is data taken from Steve Backus's lab at Stanford. Remarkably, our model provides highly accurate predictions for the spike trains of these retinal ganglion cells on new held-out natural movies that were never shown to the model to train. Its accuracy in predicting performance is almost as high as possible, set by a noise ceiling due to intrinsic stochasticity in the retina itself. Other models like linear nonlinear models, or in orange, or ganglion cells, GLM models in green, don't do as well as our deep neural network, which is shown in the purple bar to the right. What's interesting is if you look at hidden units in the hidden layers of our models, they behave a lot like bipolar cells and amacrine cells. In the middle left, you see a black trace and a purple trace. The black trace is recordings from a real bipolar cell, and the purple trace is quote-unquote recordings from the best matching hidden unit in our model. And you can see a very nice match, and the receptive fields match and quantitatively and so forth. But what's interesting is now that we have a very accurate model of the retinal response to natural movies, we asked, can it recapitulate decades worth of retinal physiology experiments on a whole bunch of structured stimuli. For example, these 8 stimuli classes in the bottom right. And remarkably, despite never having seen the stimuli, it could accurately reproduce all of these responses. Just to show you an example of how striking these responses are, here are kind of 3 examples of these nonlinear retinal responses. So for example, in the top, if you show a full field flashing grating to the retina will entrain to those full field flashes, but then if you emit a flash at the very end, the retina will fire a lot. It's as if the retina has learned something about the world, essentially its periodic structure, and when there's a violation of that periodicity, it sends a large complaint to the brain, and it works for a range of frequencies. Okay. There's the motion reversal response in the middle. So if a bar is moving to the right and then it suddenly reverses direction, the retina will complain a lot. All the retinal ganglion cells whose receptive fields are in the vicinity of the motion reversal will complain a lot. They'll send a huge volley of spikes to the brain. So it's as if the retina knows about Newton's first law of motion. Objects that are in motion tend to remain in motion. And when Newton's first law is violated, the retina sends a signal of that violation to the brain. You know, another phenomenon is when we move our eyes to the right, the entire world moves to the left on our retina, but we don't have an internal perception that the world is moving, right? So we can segregate this global motion from local motion, which does signify a local object moving in our field. So for example, on the bottom right in this full gray thing, if you have a full field grating just moving globally, both inside the circular receptive field and outside of it, the cell won't fire a lot, okay? But just above in panel A, if you have local motion that's out of phase with the global motion in red, then the same cell will fire a lot. So it can detect specifically local motion and it filters out global motion. So so the retina is acting like a predictive engine. It has important aspects of a model of the dynamics of the world, and it signals violations of that model. So these are actually they're not arcane effects. They're actually central to how presumably the retina operates. What's striking is that when we tested the same model that was only trained on natural movies, this model at the top, and we tested in all of these 8 stimuli classes, which included these 3 stimuli classes that I went in and explained, it got every single one of them right. It displayed exactly the same nonlinear responses. But now this raises, again, a profound question about the very nature of explanation in neuroscience. Why is it that this complex neural model, you know, complains when a flash is emitted, complains when a bar is reversed? These are qualitative phenomena, and they demand a qualitative, intuitive, mechanistic explanation. But how do you go into the, you know, thousands of parameters of this trained neural network and extract from that these explanations? So we would like to, and moreover, how do we extract experimentally testable hypotheses to drive the design of insightful future experiments using this complex model? So the basic idea that we followed is that for each stimulus, we developed an advanced methods in explainable artificial intelligence to carve out from this large complex circuit important subcircuits that were responsible for generating the response. So for example, in the emitted stimulus response, when there was a huge flash, we asked, you know, when that flash occurred, how do we trace backwards in time the causal flow of information through the circuit that led from stimulus to that flash? And then we could carve out a subcircuit that led to that. And we did this for a lot of these classes of stimuli. And what we found here is that for each of these classes of stimuli, we were able to take this complex model in the middle and extract very, very simple circuits that were good for explaining one of the phenomena, right? And remarkably, for 3 of these phenomena, for latency encoding, motion anticipation, motion reversal, we found subcircuits that had a lot of experimental support in the literature in previous papers, right? What's interesting is for the emitted stimulus response, nobody had written down a single model that could account for all aspects of the response. We were able to extract a very simple model involving 3 bipolar cell pathways with different latencies and different nonlinearities that when you put them together and impinge them on a ganglion cell, it yields all aspects of the emitted stimulus response. So this model makes direct predictions. If you knock out a particular type of bipolar cell, you'll have this differential effect on the emitted stimulus response and so forth. So because we can now understand this model and its response to specific stimuli, we can actually make testable predictions. And again, I should emphasize that nobody had ever written down a single model that could account for all of these stimuli, and that's what we have here. So in some sense, this work can recapitulate about 20 years of work on the retina. We have since expanded this work to primate retina and grid cells, which I'll talk about next. Okay. So the second part, we're going to derive from first principles the four most dominant retinal ganglion cell types from the structure of natural movies. So here, instead of trying to fit a complex model to a brain circuit, we're not going to fit it at all. We're going to try to derive it from first principles in order to try to attempt to answer the why question. Why is the primate retina organized the way it is? Why does it use these four cell types that it does? Okay. So, there's a lot of effort in experimental neuroscience going into cataloging cell types in the brain, including in the Brain Initiative, Chan Zuckerberg Cell Type Atlas, Allen Brain Institute, and so forth. However, there is no theory for why we have so many cell types, and we attempt to address this in the retina. And again, this is done by a very talented group of students and postdocs. Okay. So just to review the biological information, about 70% of all cell types in the primate retina are composed of midget and parasol cells, and they have the following salient properties. The midget cells have a very small receptive field in space, okay? But they're slow. The receptive field in time is slow, so they're slow integrators. The parasol cells have a large receptive field in space, and in time they integrate very, very fast. There's a lot of midget cells, and they're not very sensitive. They don't fire a lot. There's very few parasol cells, and they're very sensitive. They fire a lot in general. They have high firing rates. Okay, so the dominant question we'd like to understand is, why is space and time intertwined in this fashion? Why is a small spatial receptive field associated with slow integration and a large spatial receptive field associated with fast integration, and also these other two properties of number density and gain sensitivity? Okay, so that's a summary, just a qualitative summary of and these come in on and off flavors, so that gives you four cell types. Okay, so how can we explain this from first principles? Okay, so the strategy we followed is we asked, what if the retina was trying to optimally process natural movies? We have to look at natural movie statistics because we're trying to explain this intertwining between space and time. So here you have a natural movie, okay? We have a model of the retina that consists of a convolutional neural network. That is, there's a bunch of cells, all of the same cell type. So this is a retina that only has one cell type, right? Each of these cells has a spatiotemporal linear filter that it uses to process natural movies, right? And since it's the same cell type, it's the same filter copied across all of the cells. Okay, now we ask, how well does this model retina do in terms of processing or movies? Well, we try to linearly decode the natural movie and try to reconstruct it using an optimal linear decoder. And we measure performance as a function of the filter, right? And then we optimize the filter to try to find the best possible retina with one cell type, okay? We can compare that to another model retina that might have two cell types, right? One cell type with one spatiotemporal filter, the blue one that's on the right, and another cell type, the orange one, with another filter, and we jointly optimize both filters, okay? We'd like to ask, can you do better with two cell types, say on the right retina, than with one cell type on the left retina? And we do an apples-to-apples comparison. We hold the number of neurons in the left retina and the right retina the same, and we have the overall firing rate in the left and right retina the same. So we do an apples-to-apples comparison, and we ask, do you do better in representing the movie using two cell types than one? Now, what do we feed in for the movie? Well, this is a simple linear model of the retina, so that all that matters for what the optimal filter is the space-time power spectrum of natural movies, as that's shown in panel B, if you look at the left. So this is a heat map of the space-time power spectrum of natural movies. K represents spatial frequency, omega represents temporal frequency, and the structure of natural movies is such that it has this power law structure with two extreme tails. One tail at the bottom right at high spatial frequency and low temporal frequency, and one tail at the top left at low spatial frequency and high temporal frequency. Okay, this is the structure of natural movies. Now we can actually analytically solve for the optimal filters for 1 and 2 cell types or more cell types, and this is what we find. We find that you do better with 2 cell types than you do with 1, and why is that? It's because the 2 cell types with different filters can specialize to the 2 lobes of the power spectrum of natural movies. Okay, so if you look at the bottom right lobe, it has that corresponds to midget cells in the following sense. The spatial temporal filter will be concentrated on high spatial frequencies, so it'll be narrow in space. So it'll have a small receptive field, and therefore it's forced to concentrate on low temporal frequencies, so it's slow in time or slow to integrate. You have to have the receptive field tile space. So if the receptive field is small, in order to tile space, you need many, many cells. But if you have many, many such midget cells, then in order to not bust your firing rate budget, remember we're optimizing filters while constraining an overall firing rate. In order to not bust your firing rate budget, they can't fire very much. Okay, so that has the properties of midget cells. We'll get much more quantitative in the next slide. I'm being qualitative for now just to explain it. On the other hand, if you think about the upper left lobe, right, the parasol cell filters focus on that part of this power spectrum of natural movies. And so their filters have low spatial frequency, which means they're wide in space. And they also they're forced to focus on high temporal frequency because that's where the power in natural movies is. And so they have rapid integration or actually slightly differentiation. So because the receptive field is large, you don't need that many of them to tile space. So you can get away with few cells. And because there are not that many of them, you can have them fire a lot without busting your firing rate budget. Okay. Then we actually became quantitative. All right. Oops, this is a much smaller than. But anyways, in this figure, what you're looking at is you're looking at a comparison between the optimal retina that we find now using a nonlinear model of the retina, where each cell is modeled with an LN model. So it's a spatial temporal filter followed by a rectifying nonlinearity. And then the 4 optimal cell types that we derive on the left from simulation are very similar to off and on midget and parasol cells. So in the leftmost column, you see the spatial receptive field. In the second from the left column, you see the temporal receptive field. Okay. Now the 2 on the right, you see actually data collected from E.J. Chicholinsky's lab at Stanford, where you have actual receptive fields of midget and parasol cells. And you can see there's a really nice quantitative match between them. So what this is suggesting is that if you were to try to derive the retina as an optimal convolutional autoencoder of natural movies, and you allowed yourself 4 cell types, these are, by the way, black and white movies, and if you allowed yourself 4 cell types, you would actually get something that looks awfully a lot like the primate retina. Now, the next most dominant cell type in the primate retina is a small bistratified cell type, which actually has color, it differentially processes color. And in more recent work, unpublished work, we've been expanding this work to address natural color movies, and we find a small bistratified cell type popping out from our simulations and optimization. Okay, so this suggests that the cell type structure of the retina is dictated by the structure of natural movies, which is really interesting. Okay. So let's see. Okay, so now the third vignette is deriving a theory for the obligatory emergence of grid cells from biologically plausible neural circuits that path integrate. Okay. So as a mouse moves through a 2D arena, there are cells in the medial entorhinal cortex that fire if and only if the mouse is at the vertices of a hexagonal grid. These are the famous grid cells that got the Mosers and O'Keefe also for place cells, a Nobel Prize in physiology. But a natural question then is, well, who ordered that? Why, deep in the brain, are there these crystalline hexagonal firing fields? Why did evolution order grid cells? What might their purpose be? Okay. Cells, they don't exist at the sensory periphery. We only get low-level pitter-patter of photoreceptor activation patterns on the retina, proprioception, effort motor commands, and we have to construct through a circuit cells that have these grid cell firing fields. So a hypothesis that we followed, a function of medial entorhinal cortex is thought to be path integration, though one can debate whether or how much evidence there is for that function, but that's a common function that medial entorhinal cortex is thought to play a role in. So natural hypothesis might be any neural network trained to path integrate will create internal representations corresponding to hexagonal grids. So does the function of a network, i.e. potentially path integration, determine its structure or the structure of the firing fields of cells in the circuit? We weren't the first to pursue this hypothesis. So these two papers train neural networks to convert velocity inputs into positional outputs. And the left work got square grid cells in a square environment, and the right work got very heterogeneous cells in a square environment. So we asked, can you really get hexagonal grid cells as if you train a neural network in this fashion? Okay, and we did find it, and we found the key idea is to enforce a very simple biological constraint that firing rates are positive. So if you train a recurrent neural network on the bottom left, and you feed, well, what we did was we trained a recurrent neural network on the bottom left, and we fed it as input the velocity of a mouse moving through a square container. Okay, and we gave it training data of place field firing. So we demanded that the network generate a place field firing code. And then we just trained the recurrent weights and the feed-forward weights of this neural network to mediate that transformation. And then we examined the firing fields of these hidden units inside the neural network, and out popped very beautiful structured hexagonal grid cells. Right? So navigation plus a simple biological constraint implies hexagonal grid cells. Now, this is still, as of yet, an observation without a theory. Why does this actually happen? Okay. We actually developed a complete mathematical theory to explain why these hexagonal grid cells emerged in our work and why square or heterogeneous grid cells emerged in previous work. I can't go into the full theory in detail, but if you're interested, it's worked out in these two papers here. The basic intuition is that the neural network likes to learn the dominant Fourier modes in the place cell representation. And if you enforce a positivity on the firing rates, that introduces a constraint in panel I on which Fourier modes it can use. In particular, it can only use Fourier modes that sit on the vertices of an equilateral triangle, and summing Fourier modes whose wave vectors sit on the vertices of an equilateral triangle yields the hexagonal grid cells in panel J. Again, I don't expect you to really understand all because the theory is quite involved, but if you're interested, you can read those papers. Okay, so let me end now with the last topic, which is, this is also a little bit smaller for some reason. So we derive, and this will again just be an advertisement, we derive a simple theory of how neural circuits can recapitulate diverse psychology experiments on the development of semantic categories in infants. Okay, so this is motivated by discussions I had with my colleague in the psychology department at Stanford, James McClelland, who wrote this who with Timothy Rogers wrote this very beautiful book called Semantic Cognition. I highly recommend it. I bought it myself and read it to learn about Jay's work. And it highlights a range of really, really interesting psychology experiments revealing remarkable regularities in how infants learn semantic concepts. Okay? Just one example of this regularity is infants learn semantic concepts from a coarse to fine level of distinction. For example, early on, very early in age, after a few months, even if you control for perceptual similarity, infants can distinguish the difference between animals and plants. But you have to wait till they get a little bit older before they can discriminate different types of animals like birds and fish and different types of plants like trees and flowers. And then as they get older and older, they can make finer and finer scale perceptual discriminations or semantic discriminations. Okay? So what Jay did in a very beautiful body of work is that he trained neural networks to predict semantic properties of a set of objects, for example, different types of birds, animals, and so forth. And he watched the internal representations of these neural networks develop through training. And this was a model for infant semantic development. And what he found, so these are trajectories, a low dimensional visualization of the trajectories of hidden representations of the neural network, while the neural network is trained to learn about properties or basically answer questions about different types of animals and birds and so forth. And you can see that these trajectories in the center of this plot, the trajectories rapidly diverge. All the animals move to the upper left and all the plants move to the bottom right. Then there's a later divergence where the fish separate from the birds and a later divergence where the trees separate from the flowers. And then even later, individual animals and plants separate, right? So there's the same hierarchical unfolding of structure that occurs in the hidden representation of this neural network, similar to how they occur in infants. The first time I saw this, I was once astounded because it worked so well, and be disturbed because a simple neural network could a simple and nonlinear neural network could recapitulate what infants do at a qualitative level. Okay, so we wanted to understand how this worked, and so in a paper with Andrew Sachs and Jay McClelland, we in this PNS paper at the bottom, we developed a mathematical theory that explained why this happened. And basically what happens is coarse distinctions yield a powerful statistical signal that neural networks are capable of picking up on. Neural networks with hidden layers are capable of picking up on. And finer distinctions yield a weaker signal, and we can mathematically derive how fast various things will be learned as a function of these signals. And what you're seeing here on the right is a plot of an analytic expression for how internal representations will evolve in a one hidden layer linear network exposed to the same training data, similar training data as the nonlinear neural network on the left. And you can see that we can extract what happens. So this is really the difference between simulations and theory, right? The simulations can recapitulate the phenomena, but they don't explain why it occurs. And on the right, we have the theory that explains why it occurs, and you can get all the details in this paper. There's a similar kind of phenomena also in the grid cell work, right? Up until now, when we train the neural network and we get these hexagonal grid cells, that's an observation, but we don't understand how it works, and we need to go further to do theory of this fashion here in order to explain what happens. And something similar happens. I'll just go full steam back to something similar happens with this, with the retina, right? Once we have the complex neural circuit in the center, we still don't, even though it accurately predicts the retinal response to natural movies, we still don't understand how it works. So we have to do further theory, and in this case, algorithmic advances to extract a conceptual understanding of how this works. And this will be, I think, one of the central activities of the NIH Brain Initiative, if I go back to my first slide, as we move forward. We have large-scale recordings and stimulation in place. We have computational methods of training neural networks, either to mimic the brain or to derive it from first principles on training on a task, in place. We have advances in artificial intelligence. What we do not have yet are algorithmic methods to go from large-scale data and complex models to conceptual understanding. And moreover, we don't even have a conceptual understanding of what successful conceptual understanding would even look like. That's to be meta in some sense. And so I think as we go forward to the next decade of the Brain Initiative, these issues of what is the nature of explanation in neuroscience and how do we most efficiently arrive at it? How do we design experiments to arrive at it efficiently? How do we do analyses to arrive at it efficiently will come to the fore. And I think it'll be incredibly exciting as we deal with these thorny conceptual issues.